Good afternoon, everybody. This is a special Walking Stick Library broadcast. CRISPR-Enabled, Graphene-Based, Bio-Cyber Interface Model for In Vivo Monitoring of Non-Invasive Therapeutic Processes, Take 2. Now, why are we on in the middle of the afternoon, and why am I taking the time to make this special broadcast? Well, it's an unscheduled broadcast and I'm sure nobody's here. I'm not even running chat. The reason I'm on today is there's an old saying, you don't judge a book by its cover. If you're going to put an article on CloudHub, through your CloudHub news, and Todd and the rest of the VaxxChoice crew, I am speaking to you. If you're going to put that article up there, read it. Don't just look at the title. This particular article has a very scary title. And it was a very scary article. And I did use it to teach reading article workflow on my stream. I teach information literacy freelance because I think the whole world should know it, and the world would be a better place if it gave its info the love it deserved. But you got to give this article the love it deserves, or the hate, or whatever. So I'm going to drop my curtain here and minimize, and we're going to take a look at the article in question. Right here. CRISPR-enabled, graphene-based bio-cyber interface model for in vivo monitoring of non-invasive therapeutic processes. This is a very, 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 very scary title. Now, one of the things you want to do when you have a very, very scary title is read the article. And it's over here at IEEE Explore. And you may or may not have access to that. That is not a publicly funded database like Medline. There's not as much NSF and NIH literature in there, which tends to be freely available. So, what we want to do is look down here and start with the abstract. A lot of times you can get the sense for an article by reading the abstract, and we do a lot of abstract reading on Walking Stick Library. The problem is that this abstract is one of the more opaque ones. And yes, it's using every scare word, but you have to ask yourself, what are they really doing here? There's something with graphene in it. Are they putting the graphene inside me? Are they, it says it's not invasive. What do they mean by bio-cyber interface? I have no idea. I know you don't. Because I didn't when I read the title. And the abstract is not much help. Let's go deal with the abstract. Okay. The proposed model. Model can mean anything, folks. It can mean a physical thing. It can mean a mock-up. It can mean a bunch of them that they took out somewhere and tested. It can mean pencil and paper, just a drawing. It is inspired by CRISPR down here. What's that supposed to mean? Are they using CRISPR or aren't they? Here we have a graphene field effect transistor. Do they put it inside you? Is it sitting on a desk somewhere? And the rest of these words may mean something when may mean something in one context and not something in another. In other words, when I read through this, it is about as specific as a turd in my cat's litter pan. In fact, my cat's turds are a heck of a lot more specific. Ask my feline assistants, they will tell you. And somewhere in here they talk about a molecule of interest. Okay, that's another one I wanted to show all of you. And that would be over here. What molecule is it? I mean, you could be talking about something as simple as a glucose detector, an improved glucose detector. Where does that molecule come from? So we've got a problem here. We need the whole article. Over here, we've got how did I get that to happen? Ah, nice big one. We have IEEE. We also have some full text links. I don't know where they go, but IEEE is where you need to go. And that's not an easy one to get for a whole lot of reasons. Now, I have access, I had no problem accessing this. I've got access to where I used to work, which is Georgia State University. They have it. But if you want it, you're going to need the whole article here with all the pictures and everything else. And so for that, you're going to need an academic library. So we're going to go to Google, and I can show you how this works. Now, Todd, you're in the Bahamas, so I don't think you've got great access. But the rest of you, Mary and Dawn and Chris, one of you's in Kansas, one of you's in Idaho. If I want, I could search Idaho public universities. We have the University of Idaho. We have Boise State University. I'm going to use that as my example. You find the one that's near your home. Now, you're not going to be able to do this sitting at home. They're going to make you walk in the door due to copyright. This is Boise State, so if you're near Boise, Chris, just head on over. And if we search, we can search for library visitor policy. But before we do that, you want to make sure they have this article. And the way to do that is, we're going to need to go to the library anyway. Library. Here's the Albertsons Library. They're often named after things. Now, when you get into one of these, there's sometimes a big Discover box on the page. This one does not have that. But it does have articles and databases. Databases are where you want to be. This is made by LibGuides, which is made by SpringShare, right up here. There's a huge oligopoly out there. And I'm just going to show that to you for a minute. That's one of your oligopolies out there. It's alpha. Most places have this. Most places have this alphabetical. IEEE starts with the I, and you take a look. IEEE Explore Digital Library. They're going to have what you need. The next step for you when you do something like this is to check the visitor policy. I'm just going to go back. I'm going to search, and this place actually has a written one. So does GSU. Now, the good news, for you, Mary, and the good news for you, Chris, and I think Dawn. For those of you in Idaho and Kansas, you live in extremely generous states. You can walk right into the library, use a computer. Nobody's going to check you. You're not going to have to, I don't know if you have to sign in or check or show some form of ID, but there are guest computers. And sometimes somebody will have to log you in. Sometimes you get a login at the Unified Service Point, which is your combination reference circulation desk. Most of these libraries, you'll walk up to the big circulation desk and say, I'd like to use a guest computer. They will give you a guest ID and password, or they will tell you there's no password required. Westchester Community College, you just worked your tail over to a computer and sat down. Some places they will have to walk out and actually log you in. Some places have guest Wi-Fi. You can inquire about all of that. In this case, there is a library, there is an actual policy that is posted online. General Visitor Policy. And here we are. Albertsons Library, as part of its mission, serves the public and strives to foster engagement with the local community, particularly through collaborative efforts with teachers, school media specialists, and students. The library building is open to the public. Idaho citizens may come in and borrow books as special borrowers and access online resources. In you go. You should bring a flash drive with you or have your Google Drive open to upload the article, or Dropbox, or even your Microsoft OneDrive might work. You can inquire if OneDrive works, but have a way to take the article home with you. And you can take home the article. In some libraries, and I'm going to show you this. This is University of Kansas in Lawrence, home of the Jayhawks. You're going to want to go to the library. And we can even check and see if there's a library visitor policy. Visiting Scholars Community Access Workstations. Bingo. That's what you're interested in. You also will want to know that they have IEEE Explore, and you check their databases first. And this is just taking forever. One of the things that you also want to know is, I'm going to take you back to the library, is hours. Academic libraries have excellent hours. They are very retail, very much the original retail operation. And let's see. Albertson Library, using the library, contact us, journals, subject guides, reserve rooms, streaming media, course reserves. I don't see anything about hours. Using the library. Okay, that's our next one. And I just want to know your hours. I don't see the hours. Darn it. Okay, let's search library hours. Sometimes you have to search it. But anyway, there's hours. And you can see this on a calendar, and you'll typically see they're open until 10 o'clock at night. They're open Saturdays and Sundays. You can walk right in. They're open at 7 in the morning. This is typical for a large academic library. Open early and close late. This one didn't work. Come on, community access workstations. The Kansas University libraries are committed to serving, this is how this works at this is your guest access. Committed to serving the research needs of the general public and affiliated KU community users. The following procedures govern community access workstations in the university library by visitors. Note, use of the library catalog is not restricted. Visitors may determine KU holdings with personal devices connected to the KU guest Wi-Fi network or by asking assistance. So they also have a guest Wi-Fi. Community access workstations are configured to allow any visitor to the KU libraries and are located in most library locations. To visit the circulation desk to obtain a login PIN ticket for that location. Use the PIN ticket. It provides for 60 consecutive minutes of use. And users who want to save documents for later use are encouraged to bring a flash drive or upload them to cloud storage. You don't have to pay to print. Staff are unable to pause the 60-minute countdown. Users are logged off automatically at the end of the session. And please save your drives. Additional login tickets may be obtained based on how busy we are. So you can just get another one instead of doing an extend. We used to extend people. Wait time for a computer may vary. So do your print before or after. And this looks, again, you're pretty welcome here. It's just a question of making the time and getting in the door. And again, these places are open at night and on the weekends because students are busy on a residential campus, students are busy 24/7. So in this case, we're going to just go get the article through my login. And I just changed passwords a few days ago, so this is going to be a little unhappy. This is a little clunky here. And we're going to start the reading. Now, we're not going to read through this the way we did in the 3 days of workflow videos. I'm right here, Georgia State University. In we go. And I've done this more times than you want to know. PDF, please. And it's slow. Just give it a couple seconds. It'll come up. Now, you're going to be reading something very nasty and complicated. So, what you may want to do is think about why you are reading this, what you want to get out of this, and what you want to know. I do recommend reading the whole thing. It's important to get a feel for the language. It's important, especially, I would say this if you're writing a paper especially, but even in real life, to get a feel for the language, what's going on here. This was not the most unreadable article. It was very technical, but I've seen much less readable articles. But I would still break down what I want to know. Now, often when we have students starting out, I used to ask them, what should you know about your topic? And it's a trick question, and I would tell them it's that. It's tricky because the answer isn't that you should know any particular amount of knowledge, but that you should be honest with yourself about what you do know and don't know. In this case, that applies. How comfortable are you with mathematics? How comfortable are you with statistics? How comfortable are you with chemistry? How long has it been since you've read an academic article? How confident are you that you can suss your way through? If you're not confident, there's a number of devices we can use, and I semi-use them. I am confident. I had 8 credits of calculus in college. I had a fair amount of chemistry in college. I have revised open access chemistry workbooks because I have enough knowledge to science write in that discipline. Again, if you really feel you're at a deficit, get somebody who knows technical stuff to work with you. You may not need all that much technical stuff, and I'm going to show you why. It might be a very good idea to think, and we're going to think in writing today, but you can think this just in front of the mirror if you want. I hate this color, so we always change it. Anyone who ever watches me knows I do this for every video, so. You're going to ask, what do you want to know? What do I want to learn? This is very general. Is there a device? If there's no device, I don't have to worry about it. I can worry about what people are doing in a lab, but it's a different kind of worry if there's a device. And it's okay to worry, and it's okay to be afraid. That is the motivation that is getting you into this technical article. You're afraid maybe somebody can slip this to you. Can somebody slip me this device? Like a Mickey. And the fear of having things slip to you, believe me, I've got it too, but for different things than this. Can somebody slip me this device? And I can also ask, how invasive is this device? Does it put anything in my body? I'm going to keep pushing this up. Will it modify my genes? I'm making this smaller because we are needing more space. I hate this color. That's the color I want. I'm just doing a little cleanup in aisle 9. Okay, this looks much better. Will it modify my genes with CRISPR? And does this device have any benefits? Okay, now we can make this big enough for everybody to see. I like red. So if those are the kinds of questions I have, that's going to be my approach going in and taking notes and reading. Now, there are at least 3 ways to read an article like this. The first way is to dive in. And that's okay. You can even dive in taking notes. Again, if you're academically confident and have a little bit of technical background, and I have a little bit, I really do compared to these people, I'll work my way through. That doesn't work for a lot of people. If you find you're taking notes every sentence and doing a bunch of stuff like that, that is the time when you're going to get off this. You should get off and just read to get a feel for the language, the technical stuff, the diagrams. There's also a device called a KWL chart, which really helps. And you'll notice I worked through this. This was from the workflow video. And when I found something that I knew, I wrote it down. And if I partially knew it and wanted to know more, because I was clarifying it. Yeah, for example, I knew what in vivo meant. It meant in a live human or animal. And it didn't use DNA, it used RNA, edited RNA. And in this case, I wanted to know what kind of chemicals the model detected. Remember, it just said molecule of interest. It turned out it was looking mainly for proteins, so it's not a glucose monitor. Yeah, and again, you can read some of this. Sometimes I hit something I didn't understand at all. And again, that's like a vocabulary thing. You go out and Google it. And this explains here why they needed calculus. And this is my note. This is what we call sloppy note-taking. It's very minimalist. The notes form a sloppy pile. The only way I code them is paraphrase, which I don't do anything for. Quotes are blue and reactions are scarlet. The big takeaway here, you can code your notes by topic as well. So they will have 2 codes on them. I didn't do that when I took notes. And again, if you're taking a note every sentence or every few words, and you're not understanding what you're getting, or if you're getting really caught up in it and not understanding, again, the KWL chart read first to get a feel for the language, then come back. And you'll notice the big one here is and I don't know why these get bigger or smaller. But anyway, the big one here is, let me use the highlighter. The big one here to notice is the reactions. Reaction notes are good. You may even want to take them in the absence of anything else. And the other good thing about reaction notes is you can take pictures of, you can take notes on things that are missing. And I'll give you an example of that. Here is their schematic for how this would work. And again, this is wonderful that they do this because it looks wonderful, but there's a problem here. There's not a lot of vision. If I'm going to put something on a person as a sensor and let them, that and tie them to tubes or anything else, what advantage do I have if that person's laying on a table? I can put a tube in that person. I mean, that person is non-ambulatory. On the other hand, if I'm going to stick a wearable sensor on somebody to record medical data, this is a much better candidate. And yes, she is wearing a donut because we don't want her biting or licking it or biting it or pulling it off her body. She could scratch it off, I suppose, but she can't lift it off with her mouth and tongue. We don't want her licking and biting it off. But there she is enjoying herself on the cat tree. She'd be a great candidate. And she could walk around, use her scratching post, play with her toys, roll, assume this thing is securely stuck on her so she can roll. And she'd be as happy, she doesn't have to sit on a table in the vets with a tech and a vet holding her down because she's not thrilled with having a tube stuck in her. She's a much better candidate. So another good candidate would be a cow in a pasture. But these guys aren't seeing the possibilities of a wearable device. I don't know why this varies. But this is showing a lot of lack of vision. That sometimes tells you as much as what's there. The other great thing you can do when you're reading an article is look at the pictures. This is the device. This picture answers a lot of your questions. That's the graphene. It's outside of your body. Oh, see right here where I'm pointing? This sits on the outside. This is your skin on the inside. These are the microneedles right here. The receptors are here. They pick up whatever's circulating in your blood. They're not putting anything into you. They're looking for what's in there already. And this diagram shows you drug or molecule release biosensor signal. Signaling molecule is transported to the interstitial space. And this, at that point, when it's sitting in the liquid of your skin, these sensors pick up whatever it is that they're monitoring. And then the rest of this goes on outside your body in this wearable little device. That's the schematic. This is the diagram. So diagrams and pictures are your best friend. So back here, we have some answers. I'm getting another color here. that's sitting on your skin. And over here, can somebody slip me this device? We would know it's on the outside of my body. I could pull it off. Okay. And here we know that it puts microneedles in your body. Again, I have nothing against your fear being your fear is fairly reasonable. I said that. And in the case of my assistants, they'd probably have to shave them, which they wouldn't care for too much. But then they could go home in their little crates, or they could go home with the shaving and with me with a collection of patches that I apply in the house. They'd go home in their crates and be back in their apartment. And they could test in the comfort of their own home, which is not a bad idea. And improved patient comfort. At home monitoring. At home or in the barnyard. At home or on farm monitoring. Great for pets or small children. Great for pets, livestock, and small children. Damn, I have to keep changing the color back here. So yeah, this would definitely be a nice thing to have. A little wearable patch monitor. And does it sound like it's a good thing? Somewhat. And again, we don't know about these two questions yet. Okay, now when you read something like this, you're going to hit a lot of math. So far, this is high school math. We're adding a bunch of stuff. That's a summation. That's all it is. You've seen this in a spreadsheet. Lots of formulae. They're pretty good about telling you what each piece of this is. And this is where the molecules of interest are. This is a signaling molecule. In this case, it's a drug in a nanocarrier, but it could be something else. They're using drugs as an example. They don't really have name a specific drug in this article. That's another thing that you will also see that's missing is they don't name a drug. And then we get to the math. I recognize derivatives. This is calculus. D means derivative. T is time. Change in this over time. It's rise over run, basically, but a little fancier, if you want to think of it that way. And these, K means chemical kinetics. So there's lots of calculus and lots of chemistry. And this, it does a very nice diagram of how this works. You've got the little cells in their box up in the sensor. They've got the little receptors on the microneedles. There are receptor chemicals on the microneedles. When the molecule of interest comes by, it picks up it turns on the receptor. It goes to the little notch it turns on something, it turns on a complex in the cell. It goes to the little notch in the DNA. It proceeds to make an RNA that goes to the next box the next box, which has the graphene pad in it, on this outside externally worn sensor. Now, this is lovely, and the picture is much better than the calculus equations. All of these deal with rates of reaction and rates of flow. Yep, I don't need to know very much math to answer these questions, to answer our questions. But there's a problem here. And it's something you pick up if you've had college level biology. This little cell here does not have a ring of bacteria. It doesn't have a DNA ring. Bacteria and archaea have a DNA ring. More fancy cells, eukaryotes, like our white blood cells, have a nucleus. I don't see any of that in this diagram. Now, they could have left it out. But this leads to another question, and this would be red note territory. I'm going to make this right here for everybody to see. This would be a reaction or a commentary, and you put a page. If this was actually a real note, it would have a page number on it. This is page 304, and I'm just doing a very quick check because I get paranoid when I'm doing this. Yeah, we got a problem. That's not what kind of cells are they using? They have to keep them alive. They require different kinds of mediums, different kinds of cells. If you have Pseudomonas, it gets one kind of medium. It eats agar. It can eat agar. So we might want Pseudomonas sitting on a little piece of agar. If you want something that, does well with the sunlight, you might have some kind of chlorophyll-making algae living in your algal cell, or cyanobacteria that does photosynthesis. So then, anytime you go out for a walk in the sun, this thing gets its energy, since it's supposed to last a few days. So you probably, especially if you're going to put this on cattle or dogs, when you take your dog out for a walk, the sensor gets its food. So it doesn't say what kind of cells they are, and that's important. Cells have to eat. And again, what kind of medium would these cells? Yeah, this is super duperly important. I'm going to bold it. So this is a big issue that crops up here. It's missing details, just like it's missing details about how this might be applied. So that's one. And you can work your way through this. This symbol is equilibrium. Reactions go back and forth until they reach a steady state, depending on temperature and catalysts and a bunch of other things that are present. Finally, we get to the graphene. You'll notice it's not inside your body, it's sitting outside. And this is where the CRISPR happens. In liquid, there's no cellular suspension. It's not your cells. And that's another thing that's in this article. If you were reading this, you'd see it. Let me get back to our Miro board. So the CRISPR, the answer is no. The CRISPR happens and again. So you're not getting CRISPR'd. Nobody's modifying your genes. It's just some RNA chemical in a box on your skin. That box can be removed. You're good. And finally, we get further into the article, and you can do this by reading yourself. And again, the diagrams are wonderful. This shows how the CRISPR DNA works. And again, this is a reaction that goes to equilibrium. So it has to be able to be reversed and flushed out periodically, or you can't monitor over several days. And here they did the rates, and you can see they were able to get it to recover, to be reused again and again and again and again. Now that's important, because this means that this is a very comfy thing to wear. You don't have to keep going back to the doctor to be retested. You can just walk around and it will send out the data. And it's sitting on your skin. It can be removed again, but it's comfy. And there's one more thing. And I'll show this to you if I can. Yeah, right here. Let me see if I can find it, because it's easier to do this way. Okay, one match. Okay. In this section, we conduct a simulation to explore the response characteristics of the proposed bio-cyber interface. Specifically, we investigate how the various chemical molecules and process parameters influence output of the bio-cyber interface, assuming no amplification of the output. In particular, we consider the influence of the parameters that the designer can use to enhance the bio-cyber interface readout. The parameters include a whole bunch of different things. We solve all the differential equations using a numeric method, the fourth order Runge-Kutta method, which I had to look up. And that's all. What's missing? You know what's missing. And you can check this yourself, again, because the article is obtainable. What we've got here is somebody is slippery today. Me bad. Me really bad here. There we are. Let me get a color here from my box. Now, I don't expect that to set your mind at rest. And I did dig through the article to find there was no physical model. There were clues. I mean, the lack of thought about applications was a big one. I mean, one of the things I would love to see this model used for is if you've got a pasture out there that may have something noxious growing in it. And cows eat enough of it, it makes them sick. but it's traceable in the blood before they eat enough of it. And you want to make sure they're really not, you want to keep track of how much of it they're eating before you go in there and dig it up. Pasture's pretty big, you've got a lot of cows, maybe you want to buy them feed so they don't eat the noxious stuff, whatever. But before you invest that money, you want to check how much of it the cows are eating. And these things are cheap enough that you can buy a package of them, plop them on each cow in the barn, out go the cows. You open your Cow-O-Matic on your computer and it checks the blood, that checks the presence of alkaloid from the noxious weed in their blood. If somebody is getting in the noxious weed, you're going to know about it before they get sick. It'll save you some vet bills. It's probably cheaper to buy the cows some feed than to take them to the vet. So, but no, they're not thinking of any of those things. They're just doing the math with a lot of chemical kinetics. That's it, folks. So down here we have the answer to this question. Final question, folks. And I'm going to put this back in purple. No device and seems is the operative word here, if you are fearful. Yes, this is a device somebody could induce somebody to wear without any force. Again, for the benefits of your little kid doesn't have to sit with a tube stuck in him. He can go play in the playpen while we monitor them. Or you can put this on at home and it would be great. I mean, as a mousetrap goes, it's a nice one. They talk about building a better mousetrap. Mousetrap is a really good analogy here, actually. If you are fearful that someone could force you to wear it in the near term, I think we can lay that one at rest. If they were producing these cheaply, they could come to you with the arguments that are down here at the bottom and say, look, we just put this on the outside. It doesn't put anything in your body. You don't like the idea of wearing a wearable that monitors you. Then you're going to be talking about your rights and all that good stuff. So yeah, if you're afraid, this isn't going to completely put your fears at rest. But it's not an immediate, clear, and present fear. That's the important takeaway. And there's no physical model. And we could argue whether a nice little remote wearable for medical monitoring is a good thing. But we're not going to be buying one on the market anytime soon, and reasonable people may disagree. What reasonable people do have to do is read the article, even if it's tough. Look at the pictures, look for what's missing. If you're hitting a lot of words that you don't understand, look them up. Try to understand what you've got and understand what's in it for you. You can do this. 18-year-olds going off to college do this all the time. In fact, people who are taking AP Bio who are near university do it all the time. So this is not something that you can't do. And if you would like the article, and you really can't get it on your own, I'm going to give you my email address. I'm going to put it on the board. Drop it, let me go back over to Miro, and I will write you my email address if you really want to try reading this on your own. But when you get something that has a scary title, do yourself a very big favor. Read it. This is me, E.H. Kramer at proton.me. I'll give you a couple seconds to photograph that if you'd like. You can email me and just say you'd like you saw this video, would like a copy of the PDF. I can't mass send it out to people or post it on the web. But if you're motivated enough to ask me, I can share it with a few people. I don't think that many people stuck out this video to the end. Okay. I love putting up my curtain. Thanks for bearing with me in the middle of the day. It was great, and I'm glad we stuck it out. And the boxes in the corner of my living room that need to get broken down and either stored or tossed. They were the boxes that had all my Hamid stuff in them during Passover. All of those boxes need to get dealt with. But this was way more important than a bunch of cardboard. Todd and your crew, Todd and everybody you work with, next time you post something technical that has a scary title, do yourself and everyone out there in your audience a favor and read it first. You can do it. Don't sell yourself short. Have a great afternoon. Love your info, and I will be on at 9:30 tonight with my regular stream. Bye-bye.